Chapter 1. Bacteriology in the Victorian Era. A little more than 60 years ago, the scientific world received with almost incredulous astonishment the announcement that beer yeast consists of small spherules which have the property of multiplying, and are therefore a living and not a dead chemical substance, that they further appear to belong to the vegetable kingdom, and to be in some manner intimately connected with the process of fermentation. When Cagniard-Latour communicated the above observations on yeast to the Paris Academy of Sciences on June 12, 1837, the whole scientific world was taken by storm, so great was the novelty, boldness, and originality of the conception that these insignificant particles, hitherto reckoned as of little or no account, should be endowed with functions of such responsibility and importance as suggested by Latour. At the time when Latour sowed the first seeds of this great gospel of fermentation, started curiously almost simultaneously across the Rhine by Schwann and Kutzing, its greatest subsequent apostle and champion was but a schoolboy, exhibiting nothing more than a schoolboy's truant love of play and distaste for lessons. Louis Pasteur was only a lad of 15, buried in a little town in the provinces of France, whose peace of mind was certainly not disturbed, or likely to be, by rumors of any scientific discussion, however momentous, carried on in the great far-distant metropolis. Yet, some 30 and odd years later, there was not a country in the whole world where Pasteur's name was not known and associated with those classical investigations on fermentation, in the pursuit of which he spent so many years of his life, and which have proved of such incalculable benefit to the world of commerce as well as science. Thanks to Pasteur, we are no longer in doubt as to the nature of yeast cells. So familiar, in fact, have we become with them, that at the dawn of the 20th century we are able to select at will those particular varieties for which we have a predilection, and employ those which will produce for us the special flavor we desire in our wines or in our beers. Large and splendidly equipped laboratories exist for the express purpose of studying all kinds and descriptions of yeasts, for finding out their characteristic functions, and cultivating them with all the tenderness and care that a modern gardener bestows upon the rarest orchids. All this is now an old story, but some 60 years ago the great battle had yet to be fought which was to establish once and for all the dependence of fermentation upon life, and vanquish forever those subtle arguments which so long refused to life any participation in the work of fermentation and other closely allied phenomena. When, however, Pasteur finally cleared away the debris of misconception which had so long concealed from view the vital character of the changes associated with these processes, the bacterial ball, if we may so call it, was set rolling with a will, and information concerning these minute particles of living matter was rapidly gathered up from all directions. The recognition so long refused to bacteria was now ungrudgingly given, for it was realized at last that, in the words of M. Duclos, whenever and wherever there is decomposition of organic matter, whether it be the case of a weed or an oak, of a worm or a whale, the work is exclusively performed by infinitely small organisms. They are the important, almost the only agents of universal hygiene. They clear away more quickly than the dogs of Constantinople or the wild beasts of the desert the remains of all that has had life. They protect the living against the dead. They do more. If there are still living beings, if, since the hundreds of centuries the world has been inhabited, life continues, it is to them we owe it. Fortunately, the provisions made by nature for the preservation of the bacterial race are of so lavish a description that no fear need be entertained that this useful and indispensable world of life will be wiped out. The fabulous capacity for multiplication possessed by them, a new generation arising in considerably less than an hour, the powers of endurance which some of them exhibit in presence of the most trying vicissitudes of heat and cold, they have been known to survive exposure lasting for 7 days to a temperature of about -200° centigrade. The inability of starvation or desiccation to undermine their constitution, combine to render the question of the extinction of bacteria as remote as it is undesirable. Tempted by the prospects of exploring in this newly revealed world of life, investigators rushed into the field, and the bacterial fever has been hardly less pronounced in these last years than that rush for a material golden harvest which has characterized so many enterprises in southern latitudes. The scientific results of this microbe fever have happily, however, been of a more solid and substantial character than can be said to have followed the more tangible but sordid ventures in South African mines. Vague hypotheses have given place to facts, and bacteria have been brought more and more within the horizon of human knowledge, thanks to the genius and untiring zeal of investigators all over the world. By mechanical improvements in microscopes and subtle methods for coloring bacteria, enabling us to study their form with precision, by ingenious devices for supplying them with suitable food materials, or, in other words, by the creation of bacterial nurseries, providing the means for watching their growth and observing their distinctive habits and character, this important branch of the vegetable kingdom has been raised from obscurity to one of the principal places in our catalogue of sciences, and bacteriology has won for itself an individual footing in the scientific curriculum of our great educational institutions, and is represented in literature by such famous serials devoted to the publication of bacterial and allied researches as the Annales de l'Institut Pasteur, the Centralblatt für Bacteriologie, the Zeitschrift für Hygiene, the Annales d'Hygiène Experimentale, and other well-known journals which constitute an essential but ever-increasing burden upon the library shelves as well as pocket of the investigator. Museums of bacteria have been established, where not only specimens of particular varieties of a permanent character for comparison and reference can be obtained, but living cultivations of hundreds of different microorganisms are maintained, and only those who have had the charge of bacteria can realize the enormous amount of skilled labor involved in the catering for such a multitude, in which individual likes and dislikes in regard to diet and treatment must, if success is to be secured, be as carefully considered as is necessary in the case of the most delicate and highly pampered patient. Bacteria, by means of these depots, can in fact be bought and exchanged by collectors with as much facility as postage stamps, with the all-important difference that this collecting of bacteria is not a mere mania or speculation, but serves a most useful purpose. To the busy investigator who cannot afford either the time or space in which to maintain a large bacterial family, it is of immense convenience to be able to obtain at a moment's notice a trustworthy culture, say of typhoid or tuberculosis, or specimens of obscurer origin from air or water for purposes of investigation. These bacterial cultures are all guaranteed pure, free from contamination or admixture with other and alien microorganisms, and are strictly what they are represented to be. Although such a declaration is attached to many commodities at the present day with ludicrous incongruity, in the case of microorganisms such a breach of faith is unknown, and the antecedents of a microbe may be said to be regarded as of as much moment and to be as jealously preserved as is the pedigree of the most ambitious candidate for honours at a cattle or dog show. Amongst some of the curiosities to be found on the shelves of microbe museums may be mentioned bacteria which give out light, and thus, like glowworms, reveal themselves in the dark. These light bacteria were originally discovered in sea water and on the bodies of sea fish, and cultures of them have been successfully photographed, the only source of light being that provided by the bacilli themselves. The amount of light emitted by a single bacillus might indeed defy detection by the most sensitive plate procurable, but when gathered together in multitudes, the magnitude of which even 8 figures fail to express, these phosphorescent bacteria enable the dial of a watch to be easily read in the dark, whilst photographs of the face of a watch taken in such bacterial light have been so successful that the time at which the photograph was taken could be distinctly seen. Of bacteria it may indeed truly be said, as has Maeterlinck of the labours of bees, though it be here the infinitely little that without apparent hope adds itself to the infinitely little, though our eye with its limited vision look and see nothing, their work, halting neither by day nor by night, will advance with incredible quickness. Mention may perhaps appropriately be made here of the highly interesting fact discovered by Professor Percy Frankland, that ordinary bacteria which do not phosphoresce are capable of affecting a photographic film in absolute darkness, and can by this means produce a picture of themselves. If, however, a transparent piece of glass is placed between the bacteria and the film, no photograph results, showing that glass interferes with their activity in this respect. The author points out that as this action upon the photographic film does not take place through glass, it is in all probability due to the evolution by the bacteria of certain volatile chemical substances which either directly or indirectly enter into reaction with the sensitive film. Similar phenomena have been discovered in regard to many metals as well as organic substances, but this is the first observation which has been recorded of the action of living structures on sensitive films in the dark. We have already referred to the important services which Pasteur has rendered by distinguishing between different varieties of yeast, and separating them out according to their functions and properties. Pioneer work which has been followed up by and borne such splendid fruit in the hands of the renowned Danish investigator Emil Christian Hansen of Copenhagen. This work of isolating out individual varieties of microorganisms has been not only pursued with the energy familiar to all in the case of bacteria associated with disease, but has been pursued in various other, though perhaps less well-known, directions. A great deal of activity has lately been exhibited in so-called dairy bacteriology, and a long list has already been compiled of milk, cheese, and butter microbes, and agricultural authorities, even in this country, are slowly awakening to the fact that, in order to compete on modern lines with foreign dairy produce, dairy schools must be established where bacteriology is taught, and where instruction is given in the principles of scientific butter and cheese making. But bacteria of the brewery and of the dairy are not the only useful germs which are to be found on the shelves of microbe museums. Wine and tobacco manufacturers, on application, may respectively obtain the bacterial means of transforming the crudest must into the costliest claret, and the coarsest tobacco into the most fragrant Havana. Already considerable progress has been made in the isolation of particular varieties of wine yeast, whilst highly encouraging results have been obtained by Schussland and others in the separation of various valuable tobacco-fermenting organisms. Agricultural authorities, again, owe a debt of gratitude to these distinguished investigators, whose labours have discovered the art of imprisoning the microorganisms which play such an important part in the fertilization of the soil. Bacterial fertilizers are among the latest achievements which bacteriology has accomplished in this wonderful half-century, and the purchase of special varieties of bacteria to suit the requirements of particular kinds of leguminous plants is now fast becoming a mere everyday commercial transaction. But efforts for the amelioration of the conditions under which plant life is carried on have not been confined to providing plants with suitable bacterial friends. Vigorous and successful efforts have been made to remove from their entourage those bacterial enemies and undesirable parasites which have for so long played so important a part in the crop returns of many an agriculturalist. For the identification and separation of the plant parasites of various kinds, we have largely to acknowledge our indebtedness to American investigators, and the encouragement and support which Dr. Irwin Smith, amongst others, has received from the Government of the United States in the prosecution of these researches indicates how great is the public importance attached to them. There are in America alone 50 experiment stations where plant diseases are studied, whilst at a number of the colleges and universities more or less attention is given to the subject. Some idea of the loss occasioned to agriculturists by these plant pests may be formed by a recent announcement that the Department of Agriculture in Queensland was prepared to offer a reward of £5,000 for the discovery of a means to eradicate the prickly pear disease. Plant pathology has not yet had a distinct chair allotted to it in any of the great universities, but the subject is of such vast industrial importance that doubtless before long some seat of learning will do itself the honour to establish one, and so set the example. A striking instance of the advantages of taking stock, so to speak, of the attributes of bacteria will occur to every one in the revelation which has followed of their powers to solve one of the most knotty problems of the day, the efficient manipulation of those vast subterranean rivers of sewage which honeycomb every city of the world. The purification which sewage underwent by passing it through the pores of the soil, or in other words by filtration, was recognized about the year 1870, soon after the Rivers Pollution Commissioners had begun to make their classical investigations on the land treatment of sewage. But although the rapid transformation of ammonia into nitrates which followed the passage of the sewage through a few feet of soil was noted, yet the mechanism of this nitrification process remained a mystery until 1877, when two French chemists, Messrs. Schlossing and Münz, made the then astounding discovery that this change was dependent upon the vital energies of microorganisms. The part played by bacteria in the purification of sewage thus became an established fact, and the later experiments have been devoted to studying the necessary conditions under which the maximum amount of work is obtainable from these novel bacterial labourers. Two different classes of bacteria are required to carry on the purification of sewage: those which flourish in the absence of air and are known as anaerobic bacteria, and those to which the presence of air is essential for the exercise of their functions, the latter being therefore called aerobic bacteria. The work of the anaerobic labourers consists in breaking down the complex organic compounds present in sewage, whilst the completion of the process of purification is left to the aerobic varieties. In the ordinary course of nature both these processes are going on side by side, but it has been found advisable to separate these two different classes of bacteria as far as possible, and allot distinct premises to the anaerobic and aerobic varieties respectively engaged in the purification of sewage, for by so doing experience has shown that the work is not only more expeditiously but more efficiently carried out. Now the anaerobic bacteria are supplied along with the sewage, and the retention of their services offers practically no difficulty as long as an ample allowance of space and time is given them in which to carry on their labours. The aerobic bacteria, however, besides demanding space and time, insist upon their workshops being well ventilated, and if the supply of fresh air is in any way curtailed, they stop work entirely. Hence the ventilation of the aerobic workshops becomes a matter of primary importance if the valuable services of these labourers are to be retained. To ensure a sufficient supply of air being provided, it has been found advisable to have two or more aerobic workshops or bacteria contact beds, and the sewage is passed from one on to a second, and so on, until the purification is complete. Under proper management, the sewage should leave the works as an inodorous, almost pellucid liquid, incapable of putrefaction, which may be turned into rivers or other waterways without fear of rousing the wrath of local riparian authorities. But whilst the commercial side of bacteriology, so to speak, has made such great strides, the purely scientific applications which have been made of the facts it has furnished have by no means lagged behind. Chemists, from Pasteur downwards, have made use repeatedly of special bacteria to perform delicate operations in the laboratory which other methods have either failed to accomplish or have performed in a clumsy and less expeditious manner. There can be no doubt that, as our knowledge grows from day to day, we shall find more and more how much depends upon the work of individual bacteria, and how much importance attaches to the selection of just those varieties which are of value, and the banishment of those which are detrimental. And thus the many applications which bacteria already admit of render their easy access a matter of increasing consequence, enhancing the value of bacterial institutions such as already exist on the continent. But whilst the easy access of bacteria for experimental and scientific purposes is of great importance to the investigator, their indiscriminate distribution would equally be a source of uneasiness and danger to the community at large. Already sensational fiction has made considerable capital out of the pathogenic microbe, and with the winged aid of penny publications it does not take long for suggestions of such kinds to spread in society and assume practical shape. And whilst the administration of bacterial poisons offers comparatively but little difficulty, their identification would be a far greater problem for experts than that presented by particular chemical poisons. To cope with this danger to the public, specimens of disease germs from these bacterial depots may not be supplied to applicants unless the latter can prove to the satisfaction of the director that they are connected with responsible public institutions. In recent times, indeed, one of the most remarkable practical uses to which bacteria have been put is that of poisoning agents on a large scale, or, in other words, vermin exterminators. If this new role for bacteria becomes extended, as no doubt it will, the law for the sale of noxious drugs and preparations will also doubtless be amended to cover the distribution of bacterial poisons. It was in the year 1889 that Professor Loeffler, while experimenting with mice in his laboratory at Greifswald, discovered a microorganism which was extremely fatal to all kinds of mice. The happy idea occurred to the professor that this lethal little microbe, which he christened Bacillus typhi murium, might be turned to excellent account in combating plagues of field mice in grain fields, where the devastation committed by these voracious rodents had become in parts of Greece and Russia a serious source of loss to agriculturists. Experiments were accordingly made on a small scale to test the efficiency of this bacterial poisoner in destroying field mice, and so successful were the results that Loeffler confidently announced the possibility of keeping down these pests by distributing food material infected with these bacteria over the fields which were invaded by them. The Greek government took up the question, and Loeffler's method was applied with brilliant results. The disease was disseminated with extraordinary rapidity and severity, and the mice were readily destroyed. It is highly satisfactory to find that the character of this mouse bacillus has stood the test of time, for after a period of more than 10 years most encouraging reports concerning its efficiency still continue to be received. In one of the latest of these, drawn up by the director of the Experimental Agricultural Institute in Vienna, we read that in no less than 70% of the cases in which it was employed, it was completely successful in its work of extermination. And it is interesting to note that in a considerable number of these instances it was the domestic mouse against which its energies were directed. The rat has, however, until recently escaped the hand of the bacterial executioner, but his knell has also now been sounded in the announcement that a rat bacillus has been discovered. Considering the undesirable notoriety which these rodents have of late obtained in connection with their undoubted culpability in the dissemination of plague, this discovery, if correct, should be warmly welcomed. That there is plenty of work awaiting such a microorganism may be gathered from the fact that during the outbreak of plague in Sydney, the crusade against rats which followed led to the slaughter in 1 year of over 100,000. The discoverer of this useful member of the microbial community is Sachchenko of the University of St. Petersburg, and in his memoir he states that, whilst highly virulent as regards rats, it is quite harmless to domestic animals of various kinds. Thus cats, dogs, fowls, and pigeons, when fed with food infected with the bacillus, suffered no ill effects whatever, whilst its administration in large quantities to farm stock, such as horses, oxen, pigs, sheep, geese, and ducks, was also without result. Hence its distribution, according to its discoverer, offers no danger to other animals. This idea of employing bacteria as executioners was not original, for Pasteur had already in 1888 suggested to the Intercolonial Rabbit Commission in Australia that chicken cholera microbes should be employed for destroying the rabbits, which then, as now, are such a source of difficulty and pecuniary loss to the country. No active measures appear to have been taken, however, to carry out this suggestion, one of the principal objections raised being the undesirability of introducing a disease which was at that time believed to be a stranger to the colony. Recently the idea has been revived by Mr. Pound, the government bacteriologist at Brisbane, in consequence of his discovery that chicken cholera, far from not existing in Australia, has infested poultry yards more or less extensively for several years past, although it has only lately been accurately diagnosed as such. This chicken cholera microbe is particularly well suited for the work in question, inasmuch as, whilst extremely fatal to rabbits, it produces, like Loeffler's bacillus, no ill effect whatever on farm stock of various kinds, and is perfectly harmless to man, so that its handling by the uninitiated is not attended with any personal danger. End of Chapter 1, Part 1.